So welcome everyone to the Metrics International Forum. The forum is organized by the Meta Research Innovation Center at Stanford, or short Metrics. The center is directed by John Ioannidis and Steve Goodman, and we are a research group and fellowship program dedicated to finding ways to improve the validity and transparency of scientific research. I'm Mick Siebert, a postdoc at Metrics, and I will be today's moderator. All of our international forums are recorded and can be accessed on our website. Please mute yourself during the talk and keep your questions until the end, or write them in the chat during the presentation. Our presenter today is Elisabeth Bik. Elisabeth is a Dutch-American microbiologist who has worked for 15 years at Stanford Universities and 2 years in industry. Since 2019, she's a science integrity volunteer and consultant who scans the biometric literature for images or other data of concern. She has found over 8,000 scientific papers, and her work resulted in over 1,200 retractions and another 1,000 corrections. For her work in science communication and exposing research misconduct, she received the 2021 John Maddox Prize. So if you're ready, Elisabeth, we can start.
I am. Thank you so much, Max, for hosting me today. It's my honor and pleasure to sort of be back at Stanford for an hour or so and present my work. And I realize that some people in the audience might have heard this talk before, so I hope they will still enjoy it. Yeah, so I'm Elisabeth Bik. You can find me on X, Twitter. I'm Microbiome Digest, and I run 2 blogs, Microbiome Digest and Science Integrity Digest. And that is sort of a reflection of my older work when I worked at Stanford in the field of the human and marine mammal microbiome, so the bacteria and other microorganisms that live inside human and dolphin bodies. So I worked on dolphins. So my financial disclosure are, because I'm not employed, I'm a consultant. I work for myself, and I make money by giving talks, and I receive consulting and speaker fees. And most of my fees and consulting jobs are for publishers, for institutions and funders who are investigating specific cases of science misconduct. And I specialize in images. So most of my talk will be about figures, about images that you might find in scientific papers. I also get financial aid through Patreon, which is sort of like GoFundMe, but it's a website where people can donate small amounts of money to support makers, artists, writers, and people like me. So that allows me to be able to, do my groceries and have health insurance, which, as you know, in the US is not that cheap. I also have worked for a fraudulent company called Biomedical, and so after working at Stanford, I worked 2 years at that company. I left the company because there were rumors about fraud. And it wasn't science fraud, it was insurance fraud, double billing of people on Medicare, Medicaid. And the founders later have been charged with that and fled the US and are fugitives. So this is it's on my resume. So I want to make sure that I have this out of the way, that because I'm not hiding this. I have worked for that company. It was raided by the FBI also, so it was quite a story, and I'm hoping for a book or so or a movie to come out about this. So with that all out of the way, how did I get into what I do, which is looking at images in scientific papers? So it started first because I had found by accident that somebody had plagiarized my work. You can see this here on the left. You can see this text from an InTech open book chapter, which is sort of a predatory publisher. They publish these online free books, but a lot of those texts are plagiarized. So you can see in red the text I had written, they had stolen, and not only that, they had stolen other people's work as well, shown in different colors. So all that work that I did in the first year in 2013, I worked on plagiarism. And that led me to find a PhD thesis that had not just plagiarized my work, but that had other plagiarism that had duplicated images. You can see that on the middle and the right image here. You can see that these are photos of Western blots which are sort of a technique in molecular biology to look at protein expression. And some of you might have no idea what it is, and some of you might be very familiar with that. But I hope you can see that the photos that or the parts of the photos that I've marked with colored boxes, these red and cyan colored boxes, those look extremely similar, with the exception of the one on the bottom right, which is the same photo, but now it has been flipped, it has been mirrored. So I recognized that the same photo had been used multiple times to represent different experiments. And it seemed to be not an accident, because one of the photos was rotated or flipped. So I realized that I have some talent for finding duplications. So I always spot duplications in tiles in bathrooms or floor planks in hotel rooms, for example. And of course, when a floor is real wood or when a tile is real natural stone, you would not find repetitive elements. But when they are man-made, when there are, like laminate floors or porcelain tiles, you'll often find these repetitive patterns. So I always look for them, and you can mark them with the colored boxes, as I like to do. You can point out that some of these things are repetitive. So I thought maybe I should use this for science, this gift that I have. Or maybe it's a curse, because you once you see them, you see them. So I also realized that what made me so mad about the plagiarism and the duplicated figures was that, for me, publications, science publications, are the foundation of science. This is how we scientists communicate with each other. And also, science is about finding the truth. At least that's how I feel that science should be. As scientists, we publish our papers, but not only that, we base our research on previously published papers. We build upon other science papers. So when there's an error or misconduct in a paper, that means that other people might waste a lot of time trying to replicate those results. So sort of like bricks in a wall. If you think about every generation of scientists laying bricks on top of the previous generations by citing each other's work and building on that, if one of those publications contains an error or fraud, then part of that brick wall could be tumbling down. So traditionally, I think science has been built perhaps too much on trusting each other. When we read papers, we assume that they are true. But unfortunately, there is fraud in science. So I'll tell a little bit about that. Now, there can be many problems in scientific papers. Even after peer review and publication, there could still be problems that one might find in a scientific paper. So I've listed a bunch of them here, sort of in increasing amount of being bad. I don't think the perfect paper exists, but there can be many problems. And I feel as scientists, we have the right to point those out. We might see problems with animal ethics or human ethics, or the wrong statistical methods, p-hacking, but also duplications, plagiarism, or even AI-generated text. And so some of these are worse than others, obviously, and some of these are misconduct. So there is a whole team of detectives like myself who are scanning the scientific literature, so published papers, for problems in these papers. So we call ourselves, or other people call ourselves, sleuths, like detectives, people who sort of either anonymously or under their full name will scan the literature for problems. So I've listed a bunch of names here. These are people I have interacted with. I think there's many more, and whenever you create a list like that, you're going to forget a couple of people, of course, but you can see many names that you might know, and some of these names are pseudonyms, because of course, criticizing other people's work is not always received very well. Now, what is science misconduct? I'm sure most in the audience will be familiar with that. It's either plagiarism, falsification, which is like changing results that you've measured, or fabrication, where people make up results. There can be many other things wrong in science papers, which are usually called questionable research practices. And the line between QRP and misconduct, I think, is not always that clear, because you never know if somebody just was sloppy or if they intentionally mirrored an image and reused it. There have been, of course, many cases of misconduct in the news. We were just talking just before the talk about some more recent headlines, but I think science misconduct is now regularly in the news. And even Stanford, of course, has had some issues with investigations, as I'm sure most in the audience will be aware about. So there's many reasons why people could do misconduct. There could be, the individual fraudsters, which is usually how we think about science misconduct, where perhaps a professor in power is running a very big lab and is very demanding. And so all the people working for that person will be trying to please the professor. And especially if they're on a visa and are at risk of being sent back to their home country, they might be working really hard to please the professor, and that might mean that they might choose to do misconduct. There could be people pushing false narrative, for example, that vaccines don't work, or that vaccines cause a lot of problems and deaths in people. So we've seen some of those papers luckily also being retracted. And then there's organized science fraud. We call these scientific paper mills. These are organizations that churn out paper after paper after paper and sell authorships for people who need authorships, who are looking to boost their resume. And so this is an organized type of science fraud. And it's currently believed that paper mills are starting to make up 2 to even 10% of papers that are currently being submitted to publishers for publication. And luckily, of course, most of these are being screened and not published. So I focus on image duplication because for me, looking at images is maybe the easiest way to spot a problem in a paper. It's much harder to look at tables or line graphs and spot problems there. But with photos, you can see problems. So I have sort of 3 broad type of image duplications that you might recognize. On the left, we see an example of simple duplications where the same photo has been used twice, and you can see 2 examples, and I've marked these with red boxes and with blue boxes. 2 examples where the same photo has been reused to represent different experiments. In the middle, you see an example of a repositioned photo. You see actually 4 different photos, cells being treated with different amount of radiations, and some of these photos overlap. The 2 areas marked in green and the 2 areas marked in blue are overlapping. So we're not looking at 4 different samples. There are 3 of these photos overlapping. So that's all the same sample. And so it seems to have been done intentionally, because that's a lot of errors to make in a photo or in a figure. And on the right, you see an example of an altered, a changed photo, where the photo itself has been changed. You can see in photo A, again, this is a Western blot, you can see vertical lines, signs of splicing, but you can also see that several of these Western blot lanes appear to be duplicated, marked with blue and with red boxes. Now, the type 1 duplication, we usually assume this was an honest error. That can happen. You assemble these figures together. Maybe you're a little bit sloppy in your labeling, but that could be an honest error. While the type 3 duplication shown on the right is almost always done deliberately. And whether it's misconduct, that's another question. But there is an intention to mislead, because the original photo looked different. With the type 2 duplication, we're not always sure. I think the example in the middle is an example of a deliberately done duplication. But that's my own opinion. So just to test your own ability to spot these things, I always throw in a couple of examples. So here you see 4 different photos, 4 different experimental conditions, but 2 of these photos are identical. So it's a type 1 duplication. And this is about Alzheimer's disease. And again, I do believe this is an honest error because it's a type 1 duplication. And of course, these photos all look very similar, but you can see that these 2 photos that I've marked in with orange boxes they look identical. And this paper was actually quickly corrected. The author said, oops, we made an error. And yes, they found the original photo and corrected it. So that's a good outcome. Everybody's happy. Here's another example. This is a type 2 duplication. So you see 8 different photos, and 2 of these are overlapping. And this is, of course, a little bit harder to spot. And I'm now using software to help me find these duplications. But initially, the first 7-8 years or so, I did this by eye. And, you stare at a photo and then you suddenly see that 2 of these panels are overlapping. And again, here the author said, oh, sorry, we made an error, and corrected the paper pretty quickly. So, well, I guess a good outcome. But sometimes these duplications are a little bit more extensive, if you like. So you see here 2 examples, and I'm showing these because I think if you are a scientist, you can make an error, of course. But if you make this many errors of overlapping photos, each of these is representing something different. They should not overlap. If you make this many photographic duplications, I don't know. I believe you're either so sloppy that we cannot take any other fact or presented in these papers as facts, or you do it deliberately. But either way, I feel results like these should be retracted because it also casts doubt over all the other images in these papers. But I'm showing these 2 examples. They're from 2 different journals, to show how different publishers are responding to these duplications. So on the right, you can see that PLOS ONE retracted this paper. And I think that is the only acceptable outcome. This paper should be retracted. You cannot believe anything in this paper to be true if people are this sloppy. And so that's a good outcome, I feel. But on the left, this is an MDPI journal, Molecules, with a very similar case. They decided to allow the authors to send in a new set of figures that looked nothing like this set. And that was clean, and they issued a correction. Now, I don't know. I just feel you're giving the authors a chance to send in a new set of figures that is clean. That is, it's like allowing a person to win the Tour de France but their urine test for doping is super positive after winning the race, and then allowing them to send in a clean sample 2 weeks later. No, you won the race on doping, so you should not get a medal. I just feel this is the wrong outcome. But just to show how different publishers are in responding to these issues. Now here's a case of a type 3 duplication. So here you can see 2 photos that are clearly not overlapping. There's no overlap here, but there are problems in these photos. And these are very hard to find. I don't think I would have found any of these duplications if it wasn't for software helping me, because these photos look pretty convincing. But if you look a little bit longer and stare, and if you use software, you might find many problems in these photos. So both of these photos have duplications within the photo. The example on the right is perhaps the easiest to see. And I think usually everybody is very disturbed if I mark the outcome. So there's lots of duplications here. This is from the MD Anderson Cancer Center, well known for their big pathology department. And I'm not quite sure what they're trying to hide here. Maybe there was a crack or a gap or a hole in the issue or in the tissue that they wanted to mark. But I don't think you should Photoshop your samples like this. That just doesn't seem to be good. But here there's no action yet there's no response at all from the institution, and there's no response from the journal yet. So we'll wait and see what happens. Now, of course, Stanford has also had some issues with the president being involved in an investigation on misconduct. He was not found guilty of misconduct, but there are several papers that he published before he became the Stanford president. And one of them is this particular issue in a paper where there's an overlap between 2 different samples, 2 different photos, but also it seems that part of the photos have been rearranged and are duplicated. And So this particular paper there were other problems in this paper as well, and this paper got retracted last year. And then a more current case, this is a paper that was published in Neuron, and this will be retracted. It hasn't been retracted yet, but it has been announced that this paper will be retracted. And there, if you go to PubPeer, which is a site where we comment on these papers, you will see a very long discussion, and many people contributed and found all kinds of problems. So this figure I made myself. I found that the green signals between these two figures was duplicated. Other people had found that parts of the right image had been duplicated. Then later people found that part of the left image had been duplicated. So there seems to be a lot of duplications going on, and it's hard to imagine that this happens by accident. I don't think the microscope would make these duplications themselves. So I'm not quite sure what was the issue here. But it seems that there's been some alterations in these photos. So a lot of contribution by some of the names, as you can see on the bottom, including myself. So this was sort of a group effort where people found more and more problems as we started looking at these images in more detail. So I did this for a while. Like I said, I used to scan these by eye, but I wanted to know how often we could find duplications like these. So when I still worked at Stanford, I did this research. So this is a little bit older research published in 2016. And I scanned 20,000 papers spanning about 20 different years, spanning 40 journals and 14 publishers. And in that set of 20,000 papers, I found almost 800 papers to contain duplicated figures within the paper. Half of these we estimated to have been done intentionally. So if that's misconduct, we don't really know, but at least half of them had cases of mirroring or altered images. So we thought that is probably done intentionally. So is the prevalence of misconduct 2%? That's hard to know because, I'm looking at photos, which I can spot a duplication. But if you're smart and you move your sample a little bit further under the microscope, there would not be any duplication for me to find. So I always say we only catch the dumb fraudsters, the people who leave traces. There's many ways of doing fraud that would not leave traces. So the percent of misconduct is probably much higher than 2% because these are the ones we could catch. And if you think about tables and line graphs, it's much easier to do misconduct there. So we don't know, but I think the percentage of misconduct might be much higher than 2%. Also, journals are unfortunately a little bit slow to respond. Excuse me. So these 800 papers I reported to the journals, this was around 2015. So we're almost 10 years in, but 52% of these papers, there has not been any action taken. After 5 years, so in 2020, I looked at how many papers had been retracted or corrected. That was only 1/3. 1/3 of the papers you can see in yellow in that big pie chart on the left. 1/3 had been either corrected or retracted or expression of concern. And 2/3 of the papers, there was no action taken. And like I said, now in 2024, we're down to 52% no action taken. So people, the publishers are still, I guess, working on these things, or maybe they're somewhere in a drawer and all forgotten. So I sometimes send reminders, but I was a bit frustrated with this lack of response because once you see an error in a paper, I feel we should be able to call that out, and we should be warning perhaps other people to not put all your research hypothesis on this other particular paper. So back in 2017 or so, I started to post on PubPeer, which had just been set up. That was a new website. And PubPeer.com is a website where you can comment on papers because it's direct and you can install a plugin. And so if you do a literature search, and you can see that in the bottom, in the middle, if you do a literature search and you have the PubPeer plugin installed in your browser, you will start to see these green banners appear. And these green banners are a sign that 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 paper has received a comment. So you can click on the comment, you can see who commented on that. Most of these names are from the Tree of Life, so they're anonymous accounts, but I usually post under my full name. And you can see that these papers have comments, so you can click on it and see what a potential problem might be. And you can also join the conversation. We always alert the authors and hope that they can maybe resolve some of these issues that we're finding. So by now, I've you already heard that in the beginning, in the introduction, I found around 8,000 papers with all kinds of problems, and 7,800 of those are now on PubPeer. So most of them I'm starting to post on PubPeer. I'm a little bit behind reporting them to the journals, institutions, but journals can get also an alert if somebody commented on their paper on PubPeer. And my work has resulted in 1,200 retractions so far, and about one a day, I guess. So it's slowly rising. So switching gears a little bit, I already mentioned scientific paper mills. So I think this is a growing problem that it's going to be overwhelming, especially with AI, which is, of course, artificial intelligence, is something that is on everybody's mind and that can generate, unfortunately, a lot of fake data. So paper mills, scientific paper mills, are scammers, networks that are selling authorship. So let's say they make a fake paper and then they offer these papers, these manuscripts online. You can easily go on Facebook. You can see these advertisements, as you can see on the bottom left and bottom middle. You can easily see advertisements for paper mills offering authorship. So they make money by selling these positions, authorship positions. You want to be a first author, let's say that's 500 or 5,000, depending, of course, on the impact factor of the journal, dollars, and you can become an author on this paper. So they sell position 1, position 2, position 3. And you can just pick and choose a paper to put your name on, and even though you haven't written any of it yourself. So yeah, that is not, it's sort of misconduct, but how do we define it? They didn't write the paper for sure. And often these papers, they can be real papers, but they can also be fake papers. They can be, there's many scammers online, as you know, then they all work in slightly different ways. And so we occasionally find them, but they all have in common that they sell the authorship positions. So for example, they could work together with a special issue, with a guest editor. They could perhaps give them money, bribe them, or give them kickback in order to accept these papers. Or a paper could be accepted and they add additional authors to them in the last phase. Or it could be people just writing papers because they're good writers and they have some molecular biology knowledge, and they just write fabricated papers based on all kinds of templates. They could plagiarize or synonymize papers. And of course, nowadays they can use AI, artificial intelligence, to generate a review paper that is based on data that the AI machines will gather from the internet. Just as an example, this is what we call the tadpole paper mill. So this is a set of 600 papers that I was part of, a group of people working on these, finding them. They all had very similar titles, but what they all had in common is that they all used the same background in their Western blot. So if you focus on the background, the gray speckled background of these photos, they're all the same. All the blots have the same background. All the blots, even if they were published in different journals and in different by different authors, they all had the same background. We believe these are generated with some perhaps more primitive form of AI, where but they made the authors made the mistake, or the paper mills made the mistake of using the same background. So we could catch these, but again, we're only catching the dumb fraudsters because now they're making backgrounds that are more random, so we cannot really catch them. Here's an example. This is a completely different paper mill, perhaps multiple paper mills, where they plagiarize text written by other people, but they synonymize all the words and the expression. And this is called tortured phrases, because it leads to these weird these weird names. For example, this paper here on the right, it's about bosom malignancy. Ooh, what is that? Well, if you read the text, it's about basic parts of ladies' ruins worldwide. It reads funny. It's English, but it's funny English. It's about chest peril and bosom malignancy. And then you realize, oh, it's about breast cancer. But they synonymized all the words. So you can see how you can take a text written by somebody else and then put it in some synonymizer website, and it spits out a funny text that is undetectable by a plagiarism checker, and voila, you have a new paper. And of course, if you're very familiar with machine learning techniques, you might have spotted in the abstract there's random trees or tress, and that is random forest. So there's all these funny things. So forest becomes trees and all these funny things. And this was not discovered by myself. This is, one of these set of sleuths who found this. Guillaume Cabernac is very active, and Alexander Margazinov and Cyril Labbé, they have written this preprint about this style. And again, we can catch these because the English is funny, and you can see how people are trying to evade plagiarism. But it's nowadays with AI, of course, we cannot catch these anymore. We can only find these slightly older papers that have used these techniques. And of course, like I mentioned, there can be special issues. So MDPI and a bunch of other, like Hindawi, all these, there have been publishers who were very big in special issues where they asked the guest editor, who was not part of the regular editorial team, to issue a special issue. And then the guest editor might have just worked together with paper mills and published all these papers. So there have been many retractions nowadays based on these special issues where, for example, Hindawi, I think, retracted last year, I believe 8,000 or 9,000 papers in one big swoop, because they believed that these guest editors were actually working together with paper mills and publishing all these fake papers in big amounts. So lots of retractions based on those reasons. And then, of course, artificial intelligence, I've mentioned this a couple of times. I'm sure all of us, including myself, have, or at least I'm assuming that most of you have tried AI to see if it can generate a text. And it's so easy to use, and especially for people for whom English is not their first language, including myself, it is a very nice way to write a nice text. So I'm not against AI to write scientific papers, but currently we're in a stage where we're not yet sure how to deal with AI, and especially in writing, having students write essays. Can we allow scientists to write papers using AI? Maybe 10 years from now, that will be a very normal thing. But right now, we don't really allow that yet. But you can, if you search for AI-specific phrases like, as of my last update in January 2022, blah, 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 you can find papers that have phrases like that. Or as an AI language model, I cannot help you with that. So in some cases, you can find papers that have text like that left in. Again, we're only catching the dumb fraudsters, because if you want to fraud, if you want to use non-disclosed use of AI to help you write your paper and you're smart, you'll take that out. But if you're a little bit dumb, you leave it in, I guess. And that's how we can catch these papers. But I believe we can only catch the tip of the iceberg. So there must be many thousands and thousands of papers now being published that are written by AI. And again, maybe for a review paper, this is not a bad thing, but how many papers do we need on certain topics? Certain topics have had so many review papers, it doesn't really bring anything new to the table. So I feel this is not really how we should be doing science. Basically, we're sort of generating multiple Wikipedia pages written by AI. But yeah, you can find sometimes, and you can have some fun going Google Scholar, and you'll find many of these papers. But what worries me the most, obviously, is the ability of AI to generate fake images. I always say we humans are a very visual species. we like to see and look at photos. A photo or it didn't happen, right? This is how we used to look at photos, let's say in a news article or in some big event happened. We like to look and see photos because they're the evidence that something happened. Similarly, in science, usually photos are evidence that the data really was generated. But AI now has become so good that we cannot really tell an AI photo apart from a real photo. Look at these photos on the left, which face was made by AI. There's a really cool test in the New York Times. It's actually a photo on the right of the woman with the glasses. That photo is not real. The photo on the left, of course, they search for faces that are a little bit, unusual. But we have seen the rat with the giant penis in a science paper. That is obviously not a photo, but it's complete nonsense. There's nothing scientific useful there. But more disturbingly, we can generate photos that look pretty realistic. So photos on the right, the top row are real photos. The bottom row are AI generated. They're completely fake and there's no duplication. I cannot use my software to find these photos to be fake. So I think I'm very pessimistic. Like this, if we can use this and write papers about it, this has already been used by the bad folks, the paper mills. They're probably already generating papers en masse with lots of these fake photos. And just as an example, this is Cheshire is a person who he has disclosed his identity, so I can say it's a guy, but he has been posting these things on Twitter. Like he is able to generate fake photos based. So the photos on the left, you see these tumors. The right column is AI generated. The other photos are presumably real. And the same in the photos of the colony assay. The right two columns are created by AI using the first four as inspiration. So we cannot tell these things apart. At least I cannot look at these with my eye. Maybe some software can detect that these photos were generated differently or these parts of photos, but I think it's going to be very hard to look at which photos are fake and which ones are AI generated. And again, Cheshire is a person who is a science detective. So he does this to demonstrate that this is technically possible. And he's telling us that this is fraud. But, if you're really a fraudster, you can use this technology very easy. This is just, I believe this is Photoshop that can make these images and create fake images that are non-existent. So here's my last slide. I want to end with repeating what drives me to do this work, which is that science is about discovering the truth. But there is misconduct in science. There are people faking science for whatever reason. And I feel we need to fight this and we need to be actively looking for this. Why do people do it? Because the rewards are so high. If you fake your paper, it's going to look much better. And the results are great. You can publish in higher impact factor journals. You can boost your resume. You can find a new job. And unfortunately, the consequences are small. People who have been caught doing misconduct often are not being fired. They're still getting NIH grants. They're still being able to have funding by investors. They're still, their papers are not being retracted very often. And so the consequences are small. So, why not cheat if the chance that you are caught is so small? I've talked about the mass production of fake papers by scientific paper mills. I've talked about how AI can be used to generate fake data, and that's probably already happening on a massively large scale that we don't even know. I think we're worried about the individual fraudsters, and we're failing to see the enormous fire that is scientific paper mills. Maybe it's up to 10% of all papers now that are completely fake and not written by the authors whose names are on these papers. How are we going to deal with it? We can maybe have some discussion, but focus less on quantity and metrics and focus more on openness and rigor and reproducibility. But it's going to be tough. We cannot solve this problem easily. It takes the role of everyone, not just a sleuth here and there, but reviewers, journals, institutions, funders. Everybody needs to work together to fix this. And there's a tremendous cost of misconduct, obviously, for scientists basing their research on these flawed papers, for the science critics who might be legally threatened and sued, but also for science as a whole, because we've seen too much misinformation in the past couple of years and too many people losing their faith in science. And I really hope that you don't walk away thinking that all science is flawed, because that could be, my message could be used by people who want to discredit the science as a whole. But I am a firm fan of science, and we need science to be just better and a little bit more reliable. And only then can we solve the big problems in the world. So thank you for your attention, and happy to have a discussion about this.